Good day. I'm Max Hegblom, Editor-in-Chief of FEMS Microbiology Ecology, and it is my pleasure to welcome you to today's webinar on microbial impacts of climate change. But first, happy birthday. FEMS, the Federation of European Microbiological Societies, turns 50 today. This federation of societies got started in 1974 and is now connecting microbiologists across Europe and the world. The enthusiasm and dedication of all the microbiologists, member societies, volunteers, authors, editors, staff, readers, board members, and many more is what has made FEMS the organization that it is today. So happy 50th anniversary to all of us. The mission of FEMS is connecting, promoting, and supporting microbiology through knowledge exchange and community building for a globally sustainable future. FEMS invests in science, using the income from our journals to fund charitable activities and support our community, such as through this webinar series that enables us to highlight different topics of microbiology to a broad audience across the world. Therefore, as an author, when you publish in FEMS journals and when as a reader you use these publications, you directly support the various activities in which FEMS invests back in science. As we get going, I also want to highlight the recent editorial, Microbial Solutions Must Be Deployed Against Climate Change Catastrophe, led by Raquel Peixoto, that was co-published last week in FEMS Microbiology Ecology, the ISME Journal, Sustainable Microbiology, M Systems, and several other journals. This is a collective call from joint scientific societies, institutions, editors, and publishers requesting that the global community and governments take immediate action and decisive emergency action. A multitude of microbe-based solutions have been proposed, technologies that hold great promise and could be deployed along with other climate mitigation strategies. With this call to action in mind, our webinar today will focus on some of the roles and responses of microbes to climate change. We have three wonderful speakers who will enlighten us on the pivotal role of microbial communities in our evolving climate. Cornelia Welte from Radboud University, the Netherlands. Susanne Liebner from the German Research Center for Geosciences. And finally, Joel Costa from Georgia Institute of Technology in the US. They will explore the latest research uncovering how microbes in diverse ecosystems from urban channels to Arctic coastlines are responding to and influencing environmental change. Their talks are linked to their recent papers in FEMS Microbiology Ecology. I'm sure you will enjoy their presentations, and after the talks, we will open the session for questions and discussion. So please submit your questions via the QA link, and we will get back to all of these at the end of the session. Before we start, I also want to thank the staff of FEMS and Oxford University Press for all their work behind the scenes in making these webinars happen. So thank you very much. And with that, let's get going. So our first speaker, Cornelia, is going to talk about methanotrophic potential of the Dutch canal wall biofilms. So Cornelia, welcome.
Thank you very much. And thanks a lot, Max, for the invitation to speak and to highlight our work on canal methanotrophy. So today I would like to share with you some of the results from my research group in which we investigate methane cycling microbes. And to start off, I would like to take you to the Netherlands, where I live and also where we conducted these studies I want to present to you, to look at this beautiful fortified city of Naarden. And what you see here very nicely is that obviously there is man-made canals in this city, and these are already hundreds of years old. So humans have worked on shaping their environment in this way in a lot of cities around the globe. And at the moment, we have around half of the population living in cities that contain urban waterways. And the projections are that in the future, something like 66% of all humans are going to live in cities with urban waterways. So this is not like a niche ecosystem. It's like a really large ecosystem. And we need to know what's going on in there. And today I would like to ask the question together with you and try to answer it. What do urban canals have to do with climate change? Well, obviously, they can have to do everything with climate change because urban waterways are actually hotspots of methane emissions. And I'm sure the subsequent speakers will also talk about which ecosystems are implicated with climate change. But just to mention kind of a relation, urban waterways are thought to emit as much methane as tropical wetlands, which are huge, huge emitters. And because these ecosystems are going to be expanded and expanded, it is really important that we learn, what is living inside these ecosystems, and for the future how we can mitigate that. To put a little bit of perspective, I want to talk about the different greenhouse gases that we have in our environment. And for this, I'm showing you this chart here from from the previous IPCC report. And on the y-axis, you can see the greenhouse gas emissions normalized to gigaton CO2 equivalents. And on the x-axis, you can see the progression through time from the 1970s to more or less today. And in yellow and orange, you see the emissions caused by carbon dioxide, or the greenhouse, the global warming. So that's around 75%. Then with between 15 to 20% in light blue, you see the emissions caused by methane. And then in smaller percentages, nitrous oxide, N2O, and fluorinated gases. And my research focuses on methane as a greenhouse gas and on the microbes that are involved in the turnover of methane in the atmosphere. Methane is quite a special greenhouse gas. What I'm showing you here is a comparison of different greenhouse gases. And first, I want you to focus on the lifetime. You see that there is the first entry is for CO2, and the lifetime is between 1 and thousands of years. And why is that? So CO2 is special in the way that there are both biological removal mechanisms and geological removal mechanisms. And with biological ones, you have to think about photosynthesis in plants and microbes. And of course, this happens quite rapidly, whereas geological removal mechanisms from the atmosphere, such as carbonate weathering, take thousands of years. When we then look at methane, the lifetime in the atmosphere is actually very short. It's only 12 years. And this is one of the reasons why methane has kind of come to the center of attention for climate policies. Because if you change the methane, or if you reduce the methane emissions, you can see the results on the climate basically within a human lifetime. But of course, we also need to take care that there's no more CO2 emitted into the atmosphere, because we want a long-lasting reduction of the greenhouse effect. Then next, I would like you to have a look at the cumulative forcing over 20 years. So this says something about how strong a greenhouse gas these different greenhouse gases are. And you can see that over a 20-year time scale, methane has an 84 times higher global warming potential than CO2. So you could say it's a much, much stronger greenhouse gas than CO2. And if you look at this over a 100-year time scale, then this gap becomes a little shorter. And this just has to do with the short lifetime of methane. But this also shows for every methane molecule that you convert to CO2, you're doing something really beneficial for the climate. And if then you also manage to capture the CO2 that is produced, for instance, by microbes that oxidize methane to CO2, then you're good in the game. So coming back to the urban canals. So today I want to tell you, I want to tell you about the research that we've been doing on canals and what we learned about the methanotrophic microorganisms that live in these ecosystems. And the main person I want to highlight today is Koen Pelsma. He's a previous PhD student from my group, and he graduated end of last year. And he conducted most of the experiments that I'm going to show. And here you actually see him on a sampling trip in Amsterdam. So he's in this little boat with some coring equipment and his biogeochemistry colleagues while he's collecting samples while he's collecting samples for our studies. So why are urban canals different from other aquatic ecosystems? An urban canal is, by definition, a man-made environment. And this is indicated here, for instance, by the brick walls. We also investigated canals with wooden walls, but many of them had brick walls. Urban canals have, generally speaking, a lot of nutrient input because there is, for instance, leaf litter falling from the trees around, and also maybe some things are dumped in the canals. But also there is sometimes sewage overflow when there's heavy rainfall or storm water that brings excess nutrients into urban canals. And that's much more so than with rivers that are outside of cities. So this was kind of our starting point. We wanted to know, okay, all these nutrients that come into the urban canals, are there methanogens active that remineralize the organic matter and produce methane? And then as a result, is there maybe anaerobic oxidation of methane going on, abbreviated AOM here in the sediments, or aerobic methane oxidation at the sediment-water interface or in the water? And so we started to investigate this. Our workflow for the analysis is that we, or Kuhn in this case, we started with a sampling campaign where we collected environmental samples and measured the biogeochemistry at the sites. We then took samples back to the lab and put samples into bottles and performed incubation experiments, oftentimes with methane and either oxygen or under anoxic conditions. We performed microbial community analysis with 16S RNA sequencing and metagenomics, either from incubations, but also from the environmental samples. And we did a lot of measurements then, especially of the incubations for gas measurements. So which are the players that we investigated? So the methane cycle has two main players that are the methanogens that produce the biogenic methane from CO2 and a few other small carbon compounds. And then there are the methanotrophs that oxidize methane. And it's important to know that they oxidize 50 to 80% of the methane in the sediments before it actually reaches the environment, so in sediments and water columns. So much of the methane that is produced is already captured away by this biological methane filter and never goes into the atmosphere and contributes to climate change. And I know I'm not the only speaker today here emphasizing this, but because there's this balance of methane production and methane oxidation, we wonder a lot how climate change will kind of impact this balance, because you can imagine if the methanotrophs kind of thrive a little bit better with higher temperatures, then this balance can be, it can become imbalanced and you can have much larger methane emissions. Or maybe we are lucky and the methanotrophs are doing a little bit better, and then there will be less methane emissions if there is climate change. But yeah, we need to do more research to understand this. Who are these methanotrophs? So we can generally subdivide the methanotrophs into two different classes: the aerobic methanotrophs that need oxygen to grow, and their key enzyme is methane monooxygenase, which is the first enzyme that oxidizes the methane, and it takes an oxygen molecule and combines that to methanol. And then there are the anaerobic methanotrophs. The aerobic methanotrophs are the ones that I'm now going to present first to you. We know 4 different groups of aerobic methanotrophs: the gamma and alpha proteobacteria, Microbial, and mycobacteria. So now you can see here Kuhn while he's sampling in Amsterdam. And when we started the sampling campaign, we actually wanted to study the water column and the sediment. But when he was on the boat, he looked at the canal and he said, "Oh, look, there's actually a biofilm at the canal walls." So we also brought back the biofilm to the lab, and then took these 3 different samples: the sediment, the water from the water column, and the biofilm, and queried it for their methanotrophic potential. So in how far these samples were able to oxidize methane in the lab. And these are the results from this first paper. So on the y-axis, on the left-hand side, you see the methane oxidized in millimole methane per gram dry weight of our environmental sample per day. And this is used for the biofilm and sediment. And on the y-axis on the right-hand side, you can see it per milliliter, because this is what we used to quantify it for the water. And on the x-axis, you can see the different sampling sites. This entire study happened in Amsterdam, so all these sampling sites are different sites in the waterways of Amsterdam. And what you can see here in black in the bar graph are the initial methane oxidation rates, so the methane oxidation at the start of the incubation, and then the maximum is if we incubate them for longer time, they become a little bit more active, and you can see higher rates. But what did we learn from this experiment? So we learned that the biofilm that we hadn't even kind of thought about before we did the sampling campaign is actually super, super active in methane oxidation, much more active than the sediment and water. So we wondered a little bit further. So who's in this biofilm that is eating all the methane? So we performed some 16S rRNA amplicon sequencing, and you can see the results here from the environmental sample and the biofilm incubation. So after they had been reared in the lab for a few weeks. And already in the environmental sample, you see on the very top a green taxon that is actually methanotropic. That was quite abundant already in the environmental sample and became highly enriched also when we had the biofilm in the lab. And these are the methanomonadaceae. So that was our prime kind of methanotroph that we had in our minds when we looked at these data. So we did some metagenome sequencing and constructed a metagenome assembled genome. And I don't want you to look at all the metabolism that's going on, but just at the top part of this metabolic cartoon, where methane is oxidized to methanol by the particulate methane monooxygenase, and then it is further oxidized to formate by XOXF methanol dehydrogenase. This microbe apparently only had XOXF methanol dehydrogenase, and this is kind of special because XOXF requires lanthanides for growth and not calcium, like its isoforms. And microbes that need these lanthanides and that are dependent on those are usually just known from volcanic ecosystems where a lot of these metals occur. But here we find them in kind of a very moderate environment like Amsterdam canal walls. So we hypothesized that they would get the lanthanides from the brick walls, so that this man-made intervention would actually have repercussions on the type of microbe that we would enrich on the canal walls, or that would enrich in the biofilms on the canal walls, and that we would see in the lab. We then wondered whether what we saw in Amsterdam was particular to Amsterdam, or whether it was more widespread. And we had planned sampling trips to London and Venice and a lot of exciting cities, but then the COVID pandemic hit and Koen was not able to go to all of these places. So we went to a number of different cities in the Netherlands. So the Amsterdam data that you see here is from the other study. And then we went to Den Helder, Leiden, Middelburg, Zaandam, and Zwolle. And in this chart that I'm showing you here, you can see the different methanotrophic species that occurred in our data sets. And in this chart, so this is all from canal wall biofilms. So we did find canal wall biofilms essentially everywhere. You see that in Leiden, there's no data because we lost the DNA sample. So then we looked for the constitution of the different methanotrophs in the canal wall biofilms in all these different cities. And what you see is that everywhere except for Middelburg, there's quite an abundant microbial community consisting of methanotrophs that seems to occur in the canal wall biofilm. In Middelburg, which is very close to the sea and had canals that were brackish or were more salt water, we actually did not find methane in the water column. All the other canals emitted methane, but Middelburg didn't. And we also see that there's not so many methanotrophs there. So this is kind of congruent with what we expected from the data. When you look at who's there, and I know it's a long list, but just looking at the ones where you see kind of larger graphs for some of the sampling sites, you see there is methyloglobulus, and this was the one where I showed you the metagenome assembled genome in the previous slide. So this one was present in Amsterdam and also in all the other cities where we saw methane in the water column. And then there was a second phytotype, Cranotrix, almost at the bottom of this chart, which also we found in Amsterdam and also in all the other cities. And then there were a few methanotrophs that occurred sometimes yes, sometimes not. So they seem to be more variable. So I must say, more or less to our surprise, that this biofilm is not something that we find in only one city, and that's just very specific to this one geographical location. But at least in the Netherlands, it seems to be more widespread in different kinds of canal wall in different cities. And you see also that in every city we actually queried different sampling sites, so not just one site, and we found them repeatedly and repeatedly. So we can conclude that the methanoglobulus and the crinotrix are the main methanotrophs in canal walls, in these man-made ecosystems. But then there was something else that we observed in our data. So when we looked at the environmental data of the Amsterdam canals, and I'm now showing you the 16S RNA sequencing with archaeal primers, you should look at this blue bar that I've indicated here, which is the methanoperidonaceae, which are anaerobic methanotrophs. So we were like, oh, maybe anaerobic methanotrophs in the sediments also involved in methane cycling in urban canals. And because in this first sampling round, we actually just had gotten grab samplers and got to know the ecosystem better. And the samples, we wanted to have like a proper core and slice the cores. So we went back to Amsterdam in a second sampling trip and collected soil cores from the canals. And now we want to introduce the anaerobic methanotrophs to you because, this is the next thing that we investigated here. And I would like to introduce them to you in the order in which they were discovered. So the first that were discovered are the sulfate-dependent methanotrophs, and they consist of the anaerobic methanotrophic archaea of the different subclades 1, 2, and 3. They are very closely related to methanogens, and they also use the reverse methanogenesis pathway for methane activation, and they live in syntrophy with sulfate reducers. Then there are nitrate-dependent methanotrophs that are bacteria and that feed a little bit on the anaerobicity because they produce their own oxygen and use methane monooxygenase. Then there are nitrate-dependent methanotrophs, and those were actually the phylotypes that we found in the canals. They are also archaea and use reverse methanogenesis, and metal-dependent methanotrophs, and they are more elusive. So we went to Amsterdam and performed incubation experiments. And you can see here the different sampling sites that we chose, and the ones I'm showing you are from Dijksgracht. On the y-axis, you can see the core depth, and on the x-axis, the methane oxidation potential. And then we added different types of allergen acceptors to these bottles. And what we found was, to our surprise, that nitrate did not at all stimulate these incubations. But instead, the purple graphs, graphene oxide, showed very active methane oxidation. And graphene oxide can be used as an analog for natural organic matter. So we concluded from these experiments that probably natural organic matter in the sediments is used as an allergen acceptor by these anaerobic methanotrophs. Either they export the electrons directly to the natural organic matter, shown on the left-hand side, or they live together with other bacteria and then have some kind of electricity wiring between them. And at the same time when this was going on, another PhD student in my lab, Helene Obote, she actually worked on growing Methanoperidens archaea that we have in bioreactors on electrodes where they do extracellular allergen transfer, just as what we suggested here. So I briefly want to show this to you as well. So what you would do here is that you use these archaea that do the extracellular allergen transfer, and you put them into electrochemical systems, as you can see here. And on the right-hand side, you see the working electrode where you have the bioanode in gold, where then you would attach the microbes to that would oxidize the methane and then donate the electrons to the electrode to make some electricity. And when we look at those electrons, they are full of NME archaea. So we see here that NME archaea are actually really able to donate the electrons to the extracellular space, and then in the Amsterdam canals, maybe use natural organic matter, but like we use an electrode here. That's what you can see in the gold mesh, and in blue you see the archaea. And by now we have managed to actually grow them to very high densities on the electrodes. And now we can start studying their physiology. So this is, I just want to show this to kind of show how you can go from environmental samples to kind of a strain that you can work with in the lab and with which you can do additional experiments. So now to sum up my talk, so what did we learn in our studies of the canals of Amsterdam and other cities? So there's organic material that's coming into the urban canal that's broken down by different guilds of bacteria and archaea to methanogenic substrates. Then there are different methanogens that produce methane. And I didn't talk about it today, but actually quite some of the methane is going out of the canal via ebullition, so via bubble formation, and that's escaping biological oxidation. But then we find methanotrophs in the canal wall biofilm, mainly Crinotrix and Methanoglobulus. We find some other methanotrophs in the water column and the sediment, and we find anaerobic methanotrophs in the sediments that use natural organic matter for methane oxidation. And this is a process coupled to extracellular allergen transfer. So I hope I could show you today that urban canals harbor uncultivated methane cycling microbes, and there's a lot to be discovered. And yeah, I mean, urban canals, it's so obvious to look at that, but even that is not very well constrained. So I really think that we need more research to further constrain and combat greenhouse gas emissions. And with this, I'd like to close. I'd like to thank the members of my group, and especially Koen, who performed the majority of the experiments. And I was showing some data from Helene and Martijn towards the end on the electrochemical systems. Thank you very much.
Thank you, Cornelia. Really interesting and exciting. And I really like how you combine the physiology, biochemistry, and ecology of both the aerobic and anaerobic bacteria involved in these processes. And of course, thinking also on the applications. So thanks very much. I have a whole bunch of questions, and I see that there's quite a few in the Q&A. So we'll come back and discuss this at the end of the session. So thank you very much. So our next speaker is Susanne Liebner, who's a professor of molecular environmental microbiology at University of Potsdam and also the German Research Center for Geosciences. And her lab group is working on microbial carbon dynamics in the climate system. And today, her talk will focus on Arctic coastal thermokarst lakes and lagoons, and again, more methane oxidation. So Susanne, welcome.
Yeah, thank you very much, Max, also for the invitation to speak here. Of course, quite a task after Cornelia, but at least I think I have the cooler study side. So yeah, as you said, or as Max said, I will talk about microbial assemblages in Arctic coastal sediments with a specific focus on the process of microbial methane oxidation. And we already got a wonderful introduction to that topic by Cornelia. So I can just directly link to that. So the work that I'm presenting is largely a result of collaboration, specifically with the Alfred Wegener Institute, the permafrost section of the Alfred Wegener Institute in Potsdam. And most of the work that I'm presenting is from Xishong Yang, a former postdoc in my group. So in a larger context, the work that I'm presenting aligns with the so-called permafrost carbon feedback, which is the increase of air temperatures due to the thaw of permafrost as a consequence of climate change and caused by microbial production of greenhouse gases. And this is relevant since, as you see, there is almost double the amount of carbon in the permafrost soils or permafrost region than there is currently in the atmosphere. And also we do observe, or it is reported for the circumarctic, that permafrost is warming and it's also thawing. So meanwhile, permafrost is considered a tipping element in the climate system with all the feedback mechanisms behind. And just to give you a rough idea, it is now generally agreed on that due to the permafrost carbon feedback, there could be an additional warming of our atmosphere of up to 0.1 or of 0.1 degrees by the end of this century. This may not sound so super large, but given the ambitious climate goals that I think we still have, it's still significant. And also the permafrost carbon projections are highly uncertain still, especially due to a poor representation of methane carbon, both in terms of observational data and also of modeling, meaning that the overall increase of air temperatures due to permafrost thaw could be much larger than this number here. In the permafrost carbon feedbacks, so-called thermokarst lakes, which are thaw lakes, have a very special role because they are considered greenhouse gas hotspots. So this abrupt thaw underneath lakes is said to double the radiative forcing of the permafrost carbon feedback this century. And this is on one hand due to the mobilization of old methane that has formerly been entrapped, but it's also because these constantly unfrozen sediments underneath those lakes, which are also called taliks, are natural carbon-rich anaerobic and long-term incubators. So it is observed that there is a much larger role of microbially derived methane carbon relative to CO2 carbon. And this is really different in those taliks compared to what we call gradual thaw, which means active layer deepening. And there is a really nice review by Michiel in the Zand together with Cornelia and also myself summarizing microbial taxa processes in those thermokarst lakes. So for those that are interested, you find more in this paper. However, what has still been relatively little or poorly explored is the carbon budget and greenhouse gas production in coastal thermokarst. And this is even though, as you see from this example here from Tuktoyaktuk Peninsula, coastal Arctic areas often have a very high density of these thermokarst lakes, which in the process of coastal erosion and sea level rise can become thermokarst lagoons. And in fact, it is so that a recent study has shown that more than 50% of all the lagoons along the Pan-Arctic coast originate from thermokarst lakes. Now, one has to be honest in saying that spatially, those lagoons do not really make up a super substantial part, so area, but they are extremely interesting and fascinating natural laboratories to study how biogeochemistry and also microbiology changes along this land-to-sea transition. And there's some work out here, for example, by Maren Jenrich that looked into carbon inventories and greenhouse gas production in those lagoons. Now, the specific objectives of our work was to assess the effects of thermokarst lake to lagoon transitions on sediment biogeochemistry, on microbial assemblages, and on the fate of methane. So for that, we, together with the AVI and other colleagues, we co-organized a drilling campaign on the Bukovsky Peninsula in Siberia already in 2017 that took place in April, so we were drilling from the ice. Just generally, this Bukovsky Peninsula is characterized by a high density of thermokarst structures. So thermokarst lakes there started to evolve roughly 8,000 years ago. And what we did, we drilled into 3 water bodies. One was this Lake Goldsorvio. It's a first generation thermokarst lake. And then into this Lake Northern Polar Fox and into this what we call your Polar Fox Lagoon. As you may see, those 2 water bodies here, they stem from the same basin. So originally this was one large thermokarst lake that has been thawing, refreezing, and then it was thawing again. And at some point, this northern water body got disconnected from this more southern water body. And then roughly 2,000 years ago, this part got connected to Tixi Bay, meaning to the Arctic Ocean, via this very narrow channel that you can see here. So this lagoon is what we call a semi-closed lagoon, meaning that there is a seasonal change between brackish and freshwater discharge. It's quite a dynamic system, and due to ice build up, this whole system, which is more like you see occurring on land, gets completely disconnected here from the Arctic Ocean. So we drilled these 3 cores that you can roughly see on the right bottom side here. They were between 5 and 6 meters deep, And we sampled on site for gases and for microbial analyses and all kinds of geochemical analyses. And what we now know is that this upper part of the lagoon, roughly the upper 2.5 meters, show or were affected by seawater infiltration. And when we now look into the clustering of these samples just based on environmental parameters, it's clearly that this marine influence leads to a very, very large separation of these samples. So what you see here is the top part of the lagoon, while all the other samples, including the deeper part of the lagoon, cluster together. And interestingly, this top part of the lagoon showed only, in terms of methane concentration, only 0.4 to 5% of the methane that we found in all the other samples. So the freshwater samples, but also and the deeper part of the lagoon. And at the same time, the isotopic signature of methane here was much heavier than here, while those samples showed an oversaturation with methane. Now, when we look into the clustering of the microbial community, simply based on 16S amplicon sequencing using a universal primer, we see a very similar picture to the environmental parameters. So there's a clear separation of those uppermost lagoon samples from all the other samples. So there's a distinct marine influenced and freshwater community. And one could have also expected that we rather see site-specific clusters, so that all the water bodies would have a specific community, or that we do see a separation by depth, meaning by time, considering that it's a huge time length that we are looking here at 8,000 years and more. We were then interested in what is actually dictating or what is central to the methane concentrations, but also the isotopic signature of methane in the sediment. Is it the pore water chemistry, or is it the bulk parameters, or is it the microbial communities? So we did this structural equation modeling where we used a couple of pore water parameters like iron, sulfate, and so on. We also looked into the bulk, some bulk parameters like carbon, nitrogen, but also this carbon preference index, which is indicative for the origin and maturity of the carbon. And we used some microbial community data, specifically the abundance of methane-related taxa, but also the overall community composition. And we clearly found that the highest explanatory power comes from the pore water chemistry. So here you see highly significant correlations. But at least there was more explanatory power for the concentration and isotopic signature coming from the microbial community composition. So definitely more than from the bulk parameters. We then wanted to go a bit beyond this just community clustering, and we're interested in how the taxa co-occurrence, but also potential functional connectivity would change when these lagoons form from thermokarst lakes. So we did network analysis, and as you see overall, we retrieved these like 9 modules, which overall showed almost exclusively positive connections. And based on this, the detrended correspondence analyses shown here in one dimension, you can see the assignment of modules to the different samples. And again, we see a very strong separation based on this marine influence. And at the same time, we also see a much larger module diversity from these related to those freshwater samples. And this also suggests a higher functional diversity in those freshwater sediments and a strong habitat filtering after the lagoon formation, meaning also long before this, those sediments would propagate onto the Arctic shelf. I will now in the second part, or in the last part of my talk, focus more on the methane-related microbial community. So what you see here is kind of a zoom into those taxa that are known to produce or oxidize methane. So these like typical aerobic methane oxidizers, we heard about them already by Cornelia, and also these known anaerobic methane oxidizers, which Cornelia also already introduced. And you see their abundance by depth for these three water bodies, the two lakes and the lagoon. And what was most striking for us was this high abundance, relative abundance of so-called marine ANMEs, so these ANME 2A and B clusters, which are known to couple AOM, so anaerobic oxidation of methane, to sulfate reduction. And we also found, at least in the top centimeters of all the sediments, quite a high abundance of aerobic methanotrophs. But we think, or we are pretty confident by now, that especially those, this consortia here, or those groups, lead to a very efficient methane oxidation in the lagoon. And the reason is that when looking here on the right side into the isotopic signature of the methane carbon and also the methane concentrations, we see that in the lagoon, there is a sharp increase in the isotopic signature of the methane carbon deep down here below 2 meters, exactly where these taxa also have a high relative abundance. And this coincides with a decline of methane concentrations in approximately the same depth. And we do not see those changes in isotopic signature in the lakes or in the top, especially also not in the top part of the sediment. Furthermore, based on metagenomic sequencing, which we did for the lagoon sediment, we see a quite high abundance of genes for dissimilatory sulfate reduction in the top part of the lagoon, further supporting that at least sulfate-driven AOM plays a substantial role here and mitigates or reduces methane concentrations in the lagoon. We also, to get some more support for the amplicon data, looked into the taxonomy of MCR-ABG genes, which belong to the gene cluster of the methyl coenzyme M reductase, which is the key enzyme for methanogenesis and also for the reversal of methanogenesis. And as you see also here, we have a very relatively high abundance of genes that belong to ANME 2A, so to those marine ANMEs, but also at least substantial abundance of genes that belong to Candidatus methanopyridans, which we heard about also by Cornelia already. So from there, we were interested in what is the potential role of methanopyridans in this brackish environment, in this lagoon environment where formerly terrestrial sediment was exposed to marine, to the sea. So we constructed metagenome assembled genomes. And here I also want to promote a study that was led by Cornelia's group, because they also constructed genomes from the lagoon, but they specifically looked into the Bathyarchaeota. So for those of you who are more interested in Bathyarchaeota, I highlight this paper, so you will find more information on them in this paper from 2022. Among our MAGs, we also found some that indeed belong to methanopyridans. Mostly they had low quality, but we found one MAG with really good quality, so high completeness and low contamination. And based on this heat map, we again get a confirmation that this MAG 37, which belongs to methanopyridans, has the highest abundance in this kind of sulfate methane transition zone. So not at the top, but kind of a little bit below the top centimeters of the lagoon, where we also had the amplicon data pointing towards a high abundance of this group. And in the last slides, I just want to show you work in progress. So Xishong, he did pangenome analysis, which included this MAG 37. And one thing that we can already say is that it's very unlikely that our methanopyridans uses complex carbohydrate compounds, because it lacks all genes, for example, for carbohydrate binding modules or for polysaccharide lyases. It also seems quite unlikely that this methanopyridans is doing denitrification or even sulfate reduction, because the gene clusters are far from being complete. So our current idea is that indeed this methanopyridans couples reverse methanogenesis to the production of acetate via the Wood-Lyngdal pathway. And in order to do it, it uses an extracellular electron transfer, potentially with iron, oxidized manganese, or humic substances as terminal electron acceptors. And our methanopyridines also seems to have a special feature, which is this Rhodobacter-like nitrogen fixation complex, which could enable this methanopyridines to pump sodium out of the cell, so driving a sodium motive force, which not only gains energy, but which may also be a way to cope with this brackish environment. Since so far methanopyridines is mostly known as a freshwater organism. And this brings me to my take home. It's quite a lot of text, but I still would like to go through it. So I hope I could show you that this periodic input of marine microorganisms led to greater influence of and species gain or loss in the seawater influenced thermokarst lake sediments. Even this very highly dynamic semi-closed Arctic lagoon, which is clearly very different from typical marine sediments, can be a favorable habitat for AOM consortia. And we think that this high load of organic carbon and also terminal electron acceptors affords the coexistence of many different methanogens. I had no time to go into this, but maybe you saw it on this bubble plot where I zoomed into the methane related taxa. There were not only a lot of hydrogen-trophic methanotrophs, but also many methylotrophs. So coexistence of methanogens or many different methanogens with sulfate-reducing organisms and also several methane-oxidizing taxa. The role of methanopyridines remains still open, and this moment I have to say I also really appreciate what Cornelia showed, this combination with physiological studies. We have not yet done this, so we do not really have a good enrichment. So we can't really be sure on what this methanopyridines is doing, but at the moment we think that it couples the oxidation of methane or that it uses as terminal electron acceptors iron 3, manganese, or humic substances and couples this to a sodium motive force. And finally, thermokarst lagoons can mitigate sediment methane concentrations, especially compared to freshwater thermokarst ecosystems. And this happens even long before this system, so the lagoon sediments become part of the Arctic shelf, so become a real marine environment. Yeah, this was all from my side. So thank you very much for listening.
Thank you, Susanne. Really, really interesting and fun to also see the microbial connections between Dutch canals and lagoons in permafrost and how microbes are active and similar microbes are active across habitats. There's already a whole bunch of questions for you as well, so we'll come back to these at the end. So very much thank you. Our third speaker is Joel Koska. He's a professor in the School of Biological Sciences at Georgia Institute of Technology. And his lab works on various microbial processes that impacts Earth biogeochemical cycles. Today, he'll specifically focus on soil organic matter decomposition in anoxic peat of a sphagnum-dominated bog and what's happening in these peatlands. So, Joel.
All right, thanks, Max, and thanks to FEMS for inviting me to give this talk. Really excited to talk about the microbial ecology of peatlands, and specifically, as Max mentioned, to talk about the soil organic matter decomposition, the release of greenhouse gases, and the effects of warming on those processes. So, as you may well know, freshwater wetlands, and especially peatlands, are very important to the global carbon cycle, and they store a lot of carbon. So the areas that you see in green here are areas covered by peatlands, especially in the northern realm here. And though those areas comprise a relatively small part of the surface area of Earth, they store a lot of carbon. Somewhere around a third of terrestrial soil carbon is stored in these environments. And also freshwater wetlands, of course, as we've already heard, are an important source of methane to the atmosphere. So these are the science questions that we've been focused on in peatlands for over 10 years now. The big question is, will the carbon that's stored as these thick peat layers in the soils be released to the atmosphere as carbon dioxide or methane? And methane is, of course, important, as we've heard, because it stores a lot more heat than carbon dioxide. We also want to know, are especially microorganisms vulnerable to climate drivers, warming, and CO2 enrichment? How will these microbial communities change? And also, will ecosystem services like carbon storage, keeping that carbon in the ground, be compromised by climate? And you can boil down a lot of our questions in peatland microbiology to this slide. And that is, why do peatlands store a lot of carbon? We know that this is because primary production exceeds decomposition, but why is decomposition slow in peatlands? It could be because these are mostly present in northern areas. The peatland is cold, the soil is acidic, it's very nutrient poor and generally anoxic because these soils are saturated with water. And lastly, polyphenolic compounds are produced by the plants in peatlands, especially as you see in the center here, by these peat mosses of the genus Sphagnum, which is especially abundant in northern peatlands. And lastly, I just want to show you here on the upper left-hand corner, there's a lot of anecdotal evidence of organic matter preservation in peatlands. And this is the Tallinn Man, which is in Denmark. So there are a lot of dead bodies that fell into the peatlands, thousands of years ago, that were preserved really well. So you can see that there's some kind of organic matter preservation and inhibition of microbial decomposition there. So I want to talk about this project that I've been involved with for the past 10 years called Spruce and Peatland Responses Under Changing Environments. This is sponsored by the U.S. Department of Energy. And the science goals I've already presented is really to look at how climate drivers, temperature, and CO2 enrichment impact these high carbon ecosystems. And we've been studying peatlands in northern Minnesota. So this is very close to the border with Canada, the northernmost part of the United States. And the site is in the Marcel Experimental Forest. And as you can see from the picture here, it's a mixture of ecosystems from lakes to upland forest and wetlands that I'll be talking about. And so this shows you some pictures from the wetland up there. You can see that it's a forested wetland. So there are trees, spruce trees and larch trees there. There's also an understory of shrubs. And then if you look closely at the soil surface, you can see that the entire surface is overlain by peat mosses, again, of the genus Sphagnum. This is kind of an extreme environment for organisms. The soils are acidic. As I mentioned, there's no oxygen. The average temperature is 4 degrees Celsius. The environment is actually very low in salts as well as nutrients. And there's a paucity of electron acceptors. And I mentioned the high concentration of polyphenolics, which is generally coming from the mosses. I want to show you some pictures of this peat as well. Here you can see that there's where we're working in northern Minnesota, there's 2 to 3 meter thick peat layers there. And this peat has been laid down over the last 10 to 12,000 years. So the project, or the part of the project that I want to talk about, had these objectives, basically to investigate the role of dissolved organic matter in driving the temperature response of soil organic matter decomposition, and also to uncover the dynamics of the microbial communities that are mediating decomposition, how they're changing with warming. And so I just want to remind everyone of the classes of reactions that drive soil organic matter decomposition in wetland soil. So you can see on the left-hand side here, a soil column from the peatland. Peatlands are kind of weird because you can see that they're photosynthesizing from the top. Here's the moss layer on the top, and then the moss layer is degrading from the bottom. So you can see nice green moss here, and then degrading here down the soil column, which is mostly plant material and moss, dead moss. The reactions that then, like any freshwater wetland that are involved in soil organic matter decomposition, are hydrolysis, and depolymerization, fermentation, and then terminal decomposition by a variety of terminal electron accepting processes. And for the peatlands, as I've already mentioned, peat soils are generally, at least where we're working in these sphagnum-dominated systems, are low in terminal electron acceptors, inorganic terminal electron acceptors that Cornelia and Susanna were already talking about. Oxygen is largely absent, so you don't have much aerobic respiration going on. And so we think that methanogenesis should dominate the terminal decomposition of organic matter in the system. However, as we've already heard, organic compounds may also serve as a terminal electron acceptor. And though this idea has been around for a while, we still don't really know how important these organic terminal electron acceptors are to the overall carbon cycle in many environments. So I don't really have time to talk about all the pathways for methanogenesis, but I just wanted to make the point here that in general, the stoichiometry of methanogenesis dictates that there's a 1-to-1 ratio of carbon dioxide to methane produced by these various processes. And so if we observe in emitted gases or produced gases from peat soil a low ratio of carbon dioxide to methane, so a ratio close to 1, that should indicate that methanogenesis is driving overall organic matter degradation. If you look on the left-hand side, though, if we see a high ratio of carbon dioxide to methane, that would indicate that some other terminal electron accepting process is important to decomposition. And of course, as I mentioned, this is important because methane traps a lot more heat than carbon dioxide in the atmosphere. And what we observe in many field studies and incubation studies is that a lot more carbon dioxide than methane is coming out of these peatlands. Though they produce a substantial amount of methane, there's a lot more CO2 coming out. And so we performed microcosm incubations, as you see here, using field collected peat and pore water collected from the bog or DI water in these incubations. We manipulated temperature, electron acceptor and donor, and then we measured decomposition as greenhouse gas production, carbon dioxide and methane, and then we also looked at fermentation intermediates and microbial communities. And so this is our experimental design. You can see we also added nitrate as a potent electron acceptor to some treatments. We heated these bottles between 4 and 25 degrees Celsius, and we incubated them for 1 to 2 months and measured those parameters that I already mentioned. So this is a busy slide. I don't really want to go into this in that much detail. I just want to show you that we measured linear rates of CO2 and methane production in all of these treatments. And that rate data is summarized in this table. So what we found was that both carbon dioxide and methane production were stimulated by warming. And as you can see in the red boxes here, the ratio of carbon dioxide to methane declined with warming. So more methane was produced relative to CO2 with warming. We also saw that there was a switching in the effects of pore water. So half of the treatments got pore water from the bog and half of the treatments got DI water, as you see on the top here of the table. And those that had pore water had lower rates of gas production at the lower in situ temperatures, whereas when we warmed those soils, there was a switching, and pore water actually stimulated rates of greenhouse gas production at the higher temperatures. And here I'm showing the ratio of gases produced in these incubations over time. And what you can see is that the CO2 to methane ratio rapidly declines with time over the first 7 to 10 days. And I should mention that the colder treatments are shown in blue and the warmer treatments are shown in the warmer colors of red. And so you can see that rapid decline in ratios, which is especially occurring in the pore water. So the ratios are higher at the beginning of the incubation, indicating that other electron accepting processes besides methanogenesis are likely important and stimulated by the pore water, by some substances, likely organic matter, because there's not much inorganic material in the pore water stimulating decomposition. We also measured fermentation intermediates, and we mainly saw that acetate was present in the water from these incubations and also in pore waters in situ. And so this shows the accumulation of acetate with time in the incubations. Again, blue colors indicating the colder treatments. And what you can see here is that just as we saw a threshold in temperature in the mid-temperature ranges, with the effects of pore water on overall decomposition, we also see that affected acetate accumulation. So in the colder treatments, especially in the pore water incubations, we see that acetate accumulates to really high levels, whereas it doesn't accumulate in the warmer treatments. Now, there could be a lot at work here. Of course, acetate accumulates, could be accumulating either due to a lack of consumption or a slowing down of production rates. It's been shown at low temperatures that homoacetogenesis is favored over hydrogen trophic methanogenesis, so that could be going on here. And also it's been shown in many northern peatlands that acetoclastic methanogenesis may also be inhibited. So we think that what's going on here is that at the colder temperatures, terminal electron accepting processes are limiting overall degradation, whereas as the system warms, other upstream pathways are limiting degradation. That is either hydrolysis or fermentation reactions. We also looked at the microbial communities. This is data from 16S ribosomal RNA gene amplicon sequencing, and we show here that temperature had or explained the most variance in the microbial communities, more so than acetate or the effects of adding pore water. And this shows the taxa that are changing with warming in our incubations. You see the heat map on the left here and the relative abundance with time in the incubations of various taxa on the right. And what we saw was that fermentative organisms or known fermenters and known syntrophs especially responded to warming. So if you look on the bottom two panels here, you can see Smithella was one taxon that really was enhanced with warming, and also Centrophil aceticus was increasing in relative abundance with warming. And this is also sequence data from these incubations, and we also saw a higher relative abundance of methanogens and methanotrophs, as you would expect, since we saw more methane coming out with warming. And overall, we didn't really see a change in the absolute abundance of microorganisms using quantitative PCR as a proxy. And we've seen this also in the field that, that it's still a cold environment and they're just really not growing very, very fast in this environment, the microbes. Lastly, I'm showing data here from carbon substrate utilization in the incubations. These soils are kind of strange in that you can add a lot of juicy substrates for methanogens and get no stimulation in methanogenesis. Really, we added a lot of non-fermentable and fermentable carbon substrates to these incubations, and we saw that methane was only stimulated by the addition of citrate, whereas the other, for example, acetate, formate, methanol did not stimulate methanogenesis. So now I want to spend the rest of my time talking about the field aspects of this experiment. And this shows the giant chambers that the Department of Energy built up in this northern bog. These chambers are 8 meters by 12 meters, and they are warmed both from the air side and from soil warming. And this is a regression-based design. You can see that there are 12 chambers here. They're heated either 0 degrees above ambient, whatever it is outside, up to +9 degrees Celsius above ambient. And half of the chambers get twice atmospheric CO2. This shows you what it looks like inside of these chambers. So they're heavily instrumented, and we're lucky to have many scientists, over 100 scientists working on the same system. So we have a lot of data that we can build off of from this ecosystem. And this shows a timeline here. So heating started in 2014 and 2015. CO2 was added to the systems, and this experiment is slated to go for 10 years. We've been sampling pore water and soil from these chambers for the past 10 years. And I just wanted to show you that the data we get in the field corroborates what we see in our incubations and vice versa. That is, warming stimulates soil organic matter decomposition, largely stimulates. We see this in emissions, we see this in production rates that we measure in soils, and we also see that in pore water. We can collect pore water most easily because it doesn't involve collecting soil cores. If we took a lot of soil cores, there'd be no soil left in these chambers. So we have a problem with that. And what you see from this pore water CO2 and methane data is that that both are increasing with warming, but we're also seeing more methane produced relative to CO2 over time and with temperature treatment in the field. Wanted to show you too, just one slide on the changes in the plant communities. So we're seeing a large change in plant species composition. If you look in the upper right here, we're seeing a stimulation of primary production from shrubs, and the mosses are almost completely dying out in the warmer treatments, as you see in the lower right-hand panel here. And moss has a big effect on the chemistry and microbiology of the system, as I alluded to earlier. We've also collected quite a bit of data on the microbial communities, and I'll present just a little bit of that. We're just in the process of publishing this now. So we have a lot of genomes and metagenome assembled genomes. This shows you kind of a large family tree of the genomes that we've retrieved from the soils. And you can see that we get a dominance of Acidobacter and also Desulfobacter in this system. And as I mentioned, we have trouble taking a lot of cores because it's destructive to the chambers. So what we've been doing over the past 5 years is developing methods to sample pore water microbial communities as an indication of the effects of climate in the system. And if you look, I don't know if you can see in the upper right-hand corner, we have a lot of amplicon sequence data from this 10 years that we've been sampling up there. And what we've seen is at the DNA level anyway, all of these communities are fairly stable with treatment, with time. And so we don't see a lot of change with warming at the DNA level. So we've been developing this method, as you can see here. We filter the pore water and then extract DNA from the filters and study the microbial communities on them. And what we see is a large difference in community composition and diversity between the pore waters and the soil. This is just showing data from the methanogens and methanotrophs. And what you can see here is that there's an enrichment in methanogens and methanotrophs in the pore water relative to the peat. And there's a lot of them. 10%, up to 10% of the communities or higher in relative abundance is comprised of methanogens and methanotrophs. We also see that the pore water microbial communities appear to be more dynamic with warming. So a lot more ASVs are changing with warming than we see in the peat. So that brings me to the end here. I want to summarize by saying that greenhouse gas production is definitely stimulated by warming. We see that the peat becomes more methanogenic with warming. Methanogenesis appears to be favored over other terminal electron accepting processes. We do have evidence that dissolved organic matter in the pore water serves as a terminal electron acceptor, while also having a kind of inhibitory effect on overall decomposition. And we see a threshold in temperature in the controls of soil organic matter decomposition around the mid-temperature range of our incubations, such that terminal electron accepting processes appear to be limiting at low in situ temperatures, whereas hydrolysis and fermentation become more important at higher temperatures. And this system seems to be finely tuned to these extreme conditions. And for example, we don't see a big response of methanogens when we add substrates for methanogenesis, known substrates. And I just want to end by thanking all the people that do the work in my lab here at Georgia Tech, my students and postdocs. And this work involves a collaboration between Georgia Tech, Oak Ridge National Lab, and especially Florida State University, which generated a lot of the geochemical data that I showed you. So thank you very much for the opportunity to speak.
Thank you, Joe. Really interesting, and there's our questions coming up on this, and why don't we actually invite also Cornelia and Susanna on here, and we can go through some of the questions that are coming up. I know some of them have already been answered online. Really interesting in sort of thinking in all of these systems of this dynamics between methane production and methane oxidation, whether that's aerobic or anaerobic. This is one question that came in from an attendee, and this is about aerobic methane production. And there have been papers suggesting on that, and this is really addressed to all three of you. Any comments on whether you think there's aerobic methanogenesis or methane production in the systems, and would they play a role?
I can maybe make a start. So this is indeed an interesting development of the last 10 years or so, and I'm curious what kind of data will come out. The data that I've seen so far indicate that this is a minor process compared to methanogenesis, and especially in ecosystems where they're highly methanogenic, and I think all the ecosystems that the three of us have studied show a lot of methanogenesis potential. These alternative pathways are like in comparison much less important. But yeah, I'm curious what the others think about that.
You want to go first, Susanna?
Yeah, I just quickly want to add that at least the study that I presented, so we looked into sediment entirely. I think for at least I don't know any like oxic methane production in sediments. Of course, we know it from freshwater lakes, for example. We know it for the ocean, where it's kind of coupled to methylation. I think in lakes, as far as I know, it's coupled to penetrate or to exposure to light, so it's kind of coupled to photosynthesis. I agree with Cornelia that, or let's say definitely it exists and it probably occurs worldwide. But for the global methane budget, I think it's rather of minor importance. And in sediments, correct me if I'm wrong, but I think in sediments it has not been proven.
Yeah, I would agree with what both Susanna and Cornelia said, that it's a minor contributor to the overall methane budget. And I would say, though, one good thing that's come out of these studies is that we're realizing that methanogens are much less sensitive to oxygen than we thought before, right? That they, the methanogens, we see a lot of methanogenesis in the surface peat, for example, which is exposed to air often. The water table fluctuates a lot in peatlands. and still a lot of methanogenesis occurs in the surface. So obviously these methanogens are pretty flexible with regard to oxygen exposure. And that's the similar thing that we've seen evolve over the last decades on studies of sulfate reducers, right? That we know that they're a lot more flexible and adaptable to oxygen than we knew in the beginning.
And I think also very strong gradients that just within a couple of cell layers, you can have complete anoxia as well. And so methanogens can be still active in environments where you think of it being aerated. So question on other electron acceptors, and I think this is mainly to Cornelia, but it actually does impact the others. And this was the question of the nitrate-dependent ammonia oxidizers, and you indicate that you're not finding any nitrate. Now, that doesn't mean that there is a nitrate that is very quickly consumed, and therefore you don't measure. I'm thinking of an activated sludge plant that is continuously aerated, but if you measure oxygen in them, it's close to zero. So is this a similar situation, or do you think they're using something else?
Yeah, so we did think about this. And maybe to point out first that we were actually quite surprised that there was no nitrate in the water, because this is as such an indicator for like better quality than we would have thought of in like center of Amsterdam. So that was good news to start with. But when we did the incubation experiments, also the methanotrophy was not at all stimulated by nitrates. And if you would consider that there's cryptic, or cycling of nitrate that you actually can't see, you would expect that in an incubation experiment where you add excess of nitrates, that you would have like very quick stimulation of the metabolic process and very quick methane oxidation. This is not what we saw. So we think in the system that we had there, this is not going on because the incubations did not show nitrate-dependent methane oxidation.
Do you have any estimates on what's the relative role of aerobic versus anaerobic methane oxidation in these systems? I mean, again, they're interesting from the microbiology. Then once you start enriching for one system, of course, it doesn't go back to the environment. So do you know really what might be happening in the system?
Yeah, that's a very interesting question. And I think our data did not really address that. So I can answer these questions more from a like gut feeling perspective, rather than showing some data showing like, oh, we looked at this and this is the result. So generally speaking, aerobic methane oxidation is much quicker than anaerobic methane oxidation. And in these systems where you have very little sediments and a lot of water, aerated water, aerobic methane oxidation tends to be much, much more important than anaerobic methane oxidation. It's different in other systems, like when we look at marine sediments that are steep systems, then anaerobic methane oxidation is the predominant mechanism how methane is oxidized.
Would that be the same, Susanne, also in the thermokarst lagoons? What do you see there in terms of, I guess there it was mainly sulfate associated methane oxidizers.
Yes, so the interesting thing with this study was that in such a system, especially under so low temperatures, anaerobic methane oxidation has not really been observed. So as Cornelia said, it's very well studied and known for marine sediments, which have very stable conditions, steady state fluxes and whatsoever. In these lagoon environments, we have a very dynamic environment. So that was the first thing. It kind of, as far as I know, has been overlooked or not really well studied in those lagoon systems, especially not in the Arctic. But then in these sediments, there's hardly any oxygen. Oxygen may penetrate only the top millimeters into those sediments. So if there's methane oxidation, then we would expect it to be anaerobic methane oxidation. So in those sediments, the anaerobic oxidation may be a lot more important, or let's say aerobic methane oxidation should be of minor importance. But what I didn't really address is that those typical aerobic methanotrophs that we found based on the Amplicon data, some of them are known to, or there is a suspicion that they can also do anaerobic methane oxidation. Yeah, those canonical aerobic methanotrophs can switch if they lack oxygen, and then, for example, also use nitrate. And this is something we did not really address so far. That would certainly be also super exciting to look more into this.
Yeah. There's, Susanne, another question for you about methanoperidans in your systems. Do you want, and I guess whether it's in the same layer as the ANMI 2A types. So this is something that you could comment on as well.
Yeah, that was the question. I actually wanted to type the answer, but then I clicked the wrong button. So now I have to do it in real time. Yeah, so yeah, I had hoped I showed that they occur in the same zones. That is also something quite exciting, that these ANME 2A and B, so these classical marine ANMEs, they co-occur with methanoperidans in pretty much the same layers. Of course, we integrate over quite a distance. So normally those, let's say the classical sulfate methane transition zones is maybe a few millimeters, as far as I know. So here we integrate over a couple of centimeters at least. But at least in this integral, they co-occur, the marine and the marine ANMEs and methanoperidans.
Okay, and then there was a very early question already about fungi. And is there any role for them? And actually, I'm going to throw that to Joe, thinking, of course, of the peatlands where you have tremendous fungal mycelia. Any evidence of their involvement in methane degradation?
Yeah, methane degradation. No evidence of methane degradation. But we know that they're very important to decomposition, and we've studied their response to climate drivers and just published on that recently.
Yes, so in terms of actually degradation of the organic matter to then lead to the fermentation that then provides the acetate, hydrogen, and so on for
I don't know, I think it's kind of an emerging paradigm in wetlands in general, especially peatlands. You know, they're often, because most of the data is from upland ecosystems, we think of fungi as being really important to soil decomposition. However, I think in wetlands, because there's not much oxygen, they attenuate very rapidly with depth in the soil. So below the oxic layers, they're not that important.
Okay, no, yeah, no, and again, I think with the soil systems, and there are just so many different organisms interacting and so on, that figuring out that this is actually quite a lot of work. So a question regarding cultivation of the methanotrophs. One is the optimal conditions, and I'd like to link that with the other question earlier, which was about whether some of the cytochromes, are there any special type cytochromes or electron shuttle molecules involved in the electron transfer on the ANME organisms? And maybe you can comment on this aspect of the physiology.
Yeah, I guess this question goes to me. So these are very broad questions, and it's difficult to answer it in a concise answer. So to answer the first question, do you need specific cultivation conditions? And yes, absolutely. Your cultivation conditions determine everything. And if you take the standard cultivation conditions that are published, you're going to get the same standard taxa that everyone else has been culturing. So I think it matters a lot, but I can't give you a 2-sentence summary on how you should do the research, let's say. And then the question
on the topic.
Yeah. And then the question on the cytochromes, I find this very interesting. So this relates more to the later work where we actually enriching the methanoperidans. So the methanoperidans, compared to other ANMEs, have lots of CDAP cytochromes. They have 40, 50 CDAP cytochromes encoded in their genomes, depending on which MAG you're looking at. And many of them are differentially expressed. And we are not yet getting the one cytochrome that's it would be ideal if we have one cytochrome for iron-dependent growth, one for humic acid growth, one for manganese oxide. but it doesn't seem to be as simple as that. And I can't yet give you an answer, but I want to share that we're actively working on this and we have a few candidates, and hopefully next year we will have a few publications on that to elucidate at least some of this dark matter.
Very interesting. We'll be waiting for that.
I wanted to bring up something too with the group, if I may.
Sure.
So this is for Cornelia and Susanna. I'm wondering, in our system, like what Susanna presented for her system, we don't see a clear isotopic signal for methane oxidation or consumption of methane as you showed. We also see no amines in the anoxic layers. We do see a lot of canonical aerobic methanotrophs, and also we see that these canonical aerobic methanotrophs increase in relative abundance at soil depths where there's no oxygen. And I'm aware of the fact, at least in lakes, that there's an emerging story that, you know, these taxa that we thought were aerobic methanotrophs are also capable of anaerobic methanotrophy. So I wonder if you all have studied that and what the latest is on that from your labs.
Shall I go first? Yeah, we have studied that. I'm very interested in these aerobic, anaerobic methanotrophic bacteria. And I can share with you that from the data that we have in the lab, I don't think they can live without any oxygen, because I think they still need oxygen for activation of the methane molecule with methane monooxygenase. But we see them also in our anaerobic incubations and in our bioreactors where we cultivate anaerobic methanotrophs. We see that they really like to grow in a biofilm on the wall of the bioreactor where there's fluctuating medium levels, and they really like these like dynamic ecosystems where there is maybe trace amount of oxygen in our oxygen stripped gases that come into the reactor, and I think they will eat all the methane and then they will use other allergen receptors. In our case, nitrate, because we feed our cultures with nitrate. But I'm sure, like I think Susanna mentioned, iron. They are more allergen receptors, and they have a lot of large cytochromes also. So I'm very curious what the field will move into.
I can't really add any very educated thing here because we never really looked into anaerobic of aerobic methanotrophs. The only 50 cents I can contribute is that in our peatland work, we often also see substantial abundances of aerobic methanotrophs. And when we bring them in the lab, we also see that they're active, but the isotopic signature of the methane doesn't say anything about methane oxidation. So at least we share similar observation, if I got you right. I have no explanation to that. I don't know if it's something connected to this highly fluctuating water table and that there's some sort of recycling of the methane. And then, you know, it kind of, after it got really heavy, it gets depleted again. I know that cannot be. I have no answer to that. But we observe a similar thing and can't really explain it. So yeah, nice, interesting line of research, maybe, and specifically in peatlands.
Yeah, and there's also interesting studies coming out on microbial responses to what are called nanoxic conditions. So very nanomolar levels of oxygen, and the, physiological responses are very different. It's not the typical of what they're active under high oxygen, and they activate other gene systems. So I think there's a lot to learn about what's happening even in by aerobes, but when they are very limited in their oxygen. Okay, I think we're going to have to wrap this up. This has been a really interesting discussion and webinar. So thank you, Susanna, Cornelia, and Joe, for your wonderful presentations. Really interesting. And again, for everybody in the audience, you can read more through the links to their papers. I know we didn't get through all the questions but for the sake of time, I think we need to wrap up. So again, and thank you everybody in the audience. We actually had a great number of people, around 400 people from all around the world. So this is just wonderful of how we're able to connect. And finally, thank you to Sadie and Joe in the background who are doing a lot of work to make all of these webinars happen. So thank you, and other staff involved in making sure that our Zoom connection is working. So thank you, everyone, and we'll meet again for another webinar or in a person conference. So thank you all.
Thanks for hosting us, Max.
Yeah, thank you. Bye-bye. And happy anniversary still.
Yes, for FEMS, yes. We'll celebrate for the rest of the day. Bye.